Hi everyone, welcome back to Useful Genetics. This is lecture 3N, where we're going to explain why we see dominance between pairs of alleles. So we'll put our definition of dominance into the context of real-world alleles, considering why the pairs of alleles that we consider in genetics textbooks aren't the same as the pairs of alleles that mostly exist in the natural, genetically diverse world. We'll consider why mutant alleles are often recessive to their wild-type counterparts, and we'll consider why Mendel always saw dominance. So here's our definition. Again, we consider whether we should apply dominance when we have situations where we have two alleles that, when they're homozygous, give different phenotypes, but the heterozygote has the same phenotype as one of those two homozygotes. And we pointed out the extremely important issue that dominance describes a relationship between two alleles. It's not a property of a single allele. So if you look in genetics textbooks, even in high school genetics chapters, you will find that most of the alleles you learn about, most of the pairs of alleles you learn about, are pairs where one allele is completely dominant to the other allele. Occasionally, you'll be introduced to pairs of alleles where the heterozygote phenotype is intermediate between the two, or to pairs of alleles where both phenotypes are seen, such as the blood type alleles. What you won't learn is that in the real world, these are just the extremes of a continuum of kinds of relationships. Real alleles haven't read the genetics textbooks. They don't need to fall into discrete categories. The world of proteins and protein functions is so diverse that every possible kind of relationship occurs in the real world between real pairs of alleles. And at the end of this video, I'll address the point of, well, why do textbooks spend so much time talking about situations where we see complete dominance? If we look in the natural environment at natural genetic variation, and we ask what kinds of phenotypes do we see when we consider real pairs of alleles, we see every possible combination, as I said. The continuum is sort of full of ways that alleles interact to produce phenotypes. However, I have to qualify this a bit with a particular kind of allele. Alleles where allele pairs where one allele is functional and the other allele is non-functional. Often we'd call this a wild type allele, where one allele is functional and one allele is not functional, very often fall into the category of complete dominance or nearly complete dominance. And the question to ask is, well, why is this true? And the answer is quite simple once we understand what proteins do, what genes do, what proteins do. Defective alleles are typically recessive to functional alleles. That's what I just said. We see complete dominance in plus-minus allele pairs. What I didn't say is that we see the plus allele dominant to the minus allele. The functional allele is dominant to the non-functional allele, or the non-functional allele is recessive to the functional allele. And we see this because for many functions in the cell, in our bodies, having half the usual amount of protein is enough to get the job done. And there's a technical term for this. It's called haplosufficiency. We'll say a gene or an allele is haplosufficient is if having half of the normal functional amount, the amount you would produce in a haploid, the amount you would produce in a heterozygote with a non-functional allele, If that's enough, then we say this gene is haplosufficient, and we expect to see that defective alleles will be recessive to functional alleles. Now I'm going to apply this to the same examples we've been thinking about. So here's our blue and white morning glories, and we said that the defective W allele, the defective form of the transcription factor, is recessive to the functional form in its effect on the production of pigment in the pathway. And this kind of makes sense because we've only got half as much transcription factor binding to the promoter of the chalcone synthase gene, so we're only generating half as much material flowing through the pathway. We're only going to produce half as much pigment. What about our second example? This is a morning glory example, but this time the heterozygote had the phenotype of the wild-type homozygote. Here we did see dominance. Does this make sense? Actually, it does, but it requires a little more biochemistry than I originally explained. Chalcone synthase, this is the step that's regulated by the white allele. This is the first step of the pathway, and it often controls the rate at which material flows through the pathway. We would say that this is typically the rate limiting step. So if only half as much as this is happening, we're only going to get half as much product. Typically, most of the other steps in a pathway are not rate limiting. There's more than enough of these enzymes to handle the amount of chalcone that the first step, the regulated step, is producing. That means that even having only half as much of the normal enzyme at this step, we'll still have much more than half of the normal flux through the pathway at this step, and so we'll produce a nearly normal amount of the blue pigment. Finally, what about our blood type example where we saw dominance? Here, we only have, maybe we only have half as much of the red sugar on the heterozygote as on the homozygote. But the phenotype that we're measuring is not the amount of sugar. Our phenotype is a yes-no. Does the A antibody react to the blood? Does the B antibody react to the blood? And having half as much sugar is plenty. Oops, go back. Is plenty for the antibody to react to it. And so our test says you have type B blood just as strongly when you're a type B homozygote as when you're in a BO heterozygote. That's why we see dominance in this case. What about our cystic fibrosis phenotypes? Well, again, we see dominance in the final phenotypes. Half as much CFTR protein is enough to get the job done. So we see there's only half as much protein, but fertility and lifespan are normal because we normally make more cystic fibrosis protein than our cells really need. Half as much gets the job done quite nicely. Now, one last point. Why did Mendel only discover, why are our textbooks full of dominant alleles? Why aren't we considering more of the natural alleles? Well, Mendel explained it very clearly. He took a wide variety of alleles, of varieties of pea plants, and he did preliminary experiments to see which ones were suitable for the kind of analysis he hoped to be able to do. And he wrote that, in German with a translation here, that some characters he decided not to use in his studies because they did not permit of a sharp and certain separation. The difference was of a, well, more or less, it was hard to define. They were not suitable for the experiments because they needed to be restricted to characters that were clearly and decisively scoreable, where he could look at the P and say, this P is green, this P is yellow, not, well, it's a little bit greener than the other one, it's a little bit more yellow. So Mendel chose to do this because this was the only way he could do the groundbreaking analysis that he did. On Mendel's part, this was a brilliant decision. If he'd kept all the alleles, he would have been totally confused. Until recently, the same logic was applying in genetics research and especially in genetics textbooks. Dominant allele pairs exhibiting where one allele is dominant to the other, were so much easier to explain, so much easier to work with, that genetics researchers and the people who wrote genetics textbooks focused on them almost exclusively. It's only now that we have a lot more powerful tools that we're able to really do justice to the diversity of how alleles interact. So what have we done? We've compared dominance in textbooks to dominance in the real world, the natural genetic variation that we see, and realized that, well, textbooks are very biased towards simple dominance gene allele pairs. We've also put dominance on a solid biochemical foundation so that you can understand when we see dominance, why is it that we see dominance? And this is based on understanding what it is that the protein normally does, the product of the gene, and how the two alleles in the pair that we're comparing, how they differ and how their differences interact. And we talked about why geneticists like dominance so much, because it makes all the analysis so much simpler. Coming up next is a bit of a housekeeping lecture. It's the last lecture of Module 3. We'll talk about how geneticists discover and name genes. For you, the most important part is understanding how you can assign names to the genes and alleles that you encounter in genetics problems in ways that will make it easier to understand what's going on. I hope to see you there.